So the last speaker is Penny Boston, who's going to try to give us an overview, maybe even a synthesis of some of the fascinating targets that we've heard about already. Penny?
Thanks. Actually, what I'm going to try to do is something somewhat different, and that is because even though I thought about Mars for a long time and focused a lot of my own work thinking about caves within a rocky terrestrial planet like Mars, now that I have ascended or descended or whatever I've done to the realm of being in the scientist herding game, right? With all of our NAI teams that are working on a plethora of amazing questions, I am pretty agnostic, really, in terms of destinations. And I love all the planets, even the ones that are probably not life-bearing. So I'm not going to talk about those, though. What I really want to talk about is what are the underlying questions? How do those drive the missions? Planet X, we don't care. Where do we actually go for the most salient pathway to really interrogating those bodies in all the ways that we need to ask them questions about what they're about. Hopefully no planetary body like this will actually see a microbe this size coming towards it, because that would be the end of astrobiology as we know it. But one of the things that I think is really critical is that we have a lot of things to ask, and each one of these is equally challenging. The actual questions to ask require a great deal of thought, and many, many different bodies, including internal NASA efforts to develop these questions, go on all the time. In fact, there's an NRC panel, a committee or whatever they call it, that is meeting right now to sort of give a new set of guidance to the agency, and in fact the entire community, about where are the sweet spots, what should we be pursuing, where are the emerging fields. And this is changing very rapidly within astrobiology, because astrobiology is intrinsically massively interdisciplinary, and this is essential. It is not enough to just bring a whole bunch of folks that know different things into a room and hope that magic thought will occur. It really is part and parcel of growing and maintaining an active, interactive, fluid, intellectually fluid group of folks that are thinking about astrobiology questions. So where to go, to me, seems a more operational consideration and flows out of what we want to ask. But there are many other things. Of course, we know that there's a great deal of debate about how we look. Do we do landed missions? Do we do orbital missions looking for patterns? How do we deal with the fact that we have exoplanet efforts to look for organic life that benefits from an increasing number of items, so you can do statistical work on that, but where the actual amount of data that you get is very, very restricted to what we can actually acquire for bodies within our solar system itself. So how we look, of course, is case dependent. One of the things that I think is probably one of the hardest jobs that folks like Jim Green and others in the agency who are figuring out which missions go when is, when do we go? When do you go to a particular body? This is something that has almost a natural cadence, and perhaps it grows out of the fact that we have a very active community of teams. So you've heard a lot of folks advocating for one planetary object or another on the basis of its potential for revealing some kind of life form to us. The aggregate of that discussion bubbles to the top and hopefully provides us with a reasonable sequence to actually tackle these. Is that a perfect process? Certainly not. And we're constrained by a lot of other things. What do we do with these discoveries and uncoveries, as I think of them? A discovery is something that you are, in my framework, that you are looking for and planning to find and hoping to find. This would be something framed by a strong hypothesis. But an uncovery is something that you stumble upon in the process of doing the rest of the science that you're trying to do and the rest of the exploration. So we know that we're constrained by budgets, by political changes at the top, and by the overall strategy. But I want to put to you the notion that a lot of what we do in any scientific enterprise is also affected by our psychology. We are, after all, primates, and we have not transcended that. So the psychology of the entire community actually shifts and changes. And we like to think that that is in response to new evidence, and to some extent it is. But I also think that there are fashions in science. There are tides in science. And this is probably something that we can't avoid and shouldn't avoid, because we can use that as an impetus to go forward. And then, of course, idiosyncrasies. We don't know what the universe is going to throw at us, and we're always alert for new twists on things that may make us rethink our strategy. One of the things that I think is really critical, and I've thought about a lot for a very long time, as have many people, and that is, what are the lessons that we can learn from our very first attempt to go look for life on another planet? Of course, this was many decades ago now, 4 decades plus, and it was a mission set that was filled with a great deal of hope, right? People were very invested in this opportunity to actually go to one of our nearest neighbors and actually look for life forms in the fashion that we then understood. And we did that, packaged it up, sent it to Mars, and nobody seemed to be home. And this was a really big bummer, right? This is one of my favorite pictures of a sad alien. And this really put a cloud over the whole idea of targeted missions. We are so much more sophisticated now that maybe we think that we're more sophisticated than we are, right? So we're not really over the lessons that we should be learning from Viking. We really need to tap back into that to think about it on a frequent basis. People have suggested that we were too soon, that maybe we should have waited until we knew more before we sent a mission like Viking. I think that that is absolutely wrong. And part of the reason is because each individual question that you ask about any aspect of science has an arc of maturation. It has the exploration phase, it has the development phase, and it has a mature phase. And the way that you approach that cannot necessarily be foreseen in advance. So I guess I'm making a plea for an individual analysis of all of these different types of questions within astrobiology that we want to ask, and that all of them are not the same. All of them will not have the same maturation arc. And when you put them all together, you get a maturation arc for the entire discipline. I would submit that astrobiology is still very much in its infancy, even though it's a number of decades old. We are still very much in a discovery phase. Part of that is because we are in the intellectual discovery phase, but part of it is also because we are so dependent on a high degree of technology to enable us to actually do missions to other planets, that we are burdened by that, as well as we are graced by that. And so those two things work together, and it's going to be a very long time before astrobiology becomes what I would think of as a mature field. In fact, it may await the fact, or the point at which we actually find some evidence of other life forms. So I just want to run down this list. At the time Viking was designed, we didn't know this long list of things. We had never heard of the deep sea hydrothermal vents. They hadn't even been discovered. We didn't know that microorganisms were living in Antarctic sandstones, or that the entire subsurface down to perhaps 10 kilometers is shot through with microorganisms. We didn't know any of that. We didn't know that a lot of microorganisms were eating their way through rock. We were right before the time at which we could look at the genomics and viromics and all of this, proteomics of living material. We didn't know there were any volcanic caves on Mars. Okay? We do now. we didn't have much of an idea that microorganisms can be included in geological materials and be quiescent or dormant for long periods of time. The degree to which that's true, we don't yet know. For my own work, it's certainly tens of thousands of years. Some people have even implied that it's millions of years. So this means that there are hidden properties within our own system that we don't even know. We think of microbes as a certain size range here, but now we understand that there are living microbes that are not starved, but just exquisitely adapted to subsurface conditions or surface conditions where they have very little energy and they're very, very small by nature. And they can be very, very slow. And so these are challenges for us to think about, trying to get out of our own heads and our own biosphere. So what happened after the Viking mission? After the period of being bummed out and sulking, the community got over it, and it resulted in an explosion of things. Extreme environment biology on this planet went wild, methodological developments all over the place, some of them as a result of the space program, some of them a result of the other things that humans are interested in, including medicine. Mars understanding with the missions that have been going to that planet, particularly for the last 20 years when Mars has sort of undergone its secondary renaissance, have shown us so much more about that particular planet. And at the same time, there's been this explosion of geomicrobiology that melds Earth science, as we understand it on this planet, with the life that lives on Earth and has co-evolved, really, with the planetary processes themselves. So one of the things that I think is potentially a problem for us is that there really is a mania for what's known as hypothesis-driven science. And Alfonso Davila just discussed some of that aspect. I think that you have to use sense. And that is that when you are looking at a very difficult and largely unconstrained problem like another planet, that you have an entire spectrum from open-ended exploration, which is perceived as high risk, but potentially high reward, and relatively unconstrained, all the way to the circumstance when you know a lot about a particular thing, and then you can form really crisp, clearly defined hypotheses that are subject to strong inference, where you can do experiments and observations that can really give you a yes or no answer to a particular question. Planetary science is across this spectrum, and so I would say in spades is astrobiology, even more so. A balanced portfolio of allowing for exploration, while at the same time, where it makes sense, focusing on highly defined, crisp, hypothesis-driven science is the only sensible way to approach it. So I just want to show one picture from my own work, which really looks at some of the more spectacular looking environments that I've worked in. And thinking about it the other day as I was putting this talk together, I thought, where in my own work have we been mostly open-ended and exploration-driven, and where have we gone into this with a specific hypothesis? And I have to say that the two cases of exploration have been probably as productive or more productive in terms of total amount of science than the cases in which we had a strong hypothesis. So for example, in the case of NICA, our hypothesis was that the fluid inclusions within the giant crystals would actually contain at least DNA and maybe other remnants of life. And what we found was not only do they contain those, but they also contain some still viable organisms. On the other hand, the work that we've done in the top system, which David showed that same picture of earlier, we went in there not knowing what we were doing. We knew it was potentially a dangerous environment. It nearly tried to kill us a bunch of times. We learned about that, and yet far more papers, of course, have come out of that top one. And this is because this is an interaction of the exploration that is unconstrained, but then turning it into hypothesis-driven science as you get more sophisticated in an environment. And I would submit that we are still largely within the exploration phase, when we are exporting our astrobiology aspirations to other bodies. So I'm not going to grind through these. I just want to say that we have a lot of very high-level questions in astrobiology that are derived both internally from NASA policy, from our NRC guidance, and other guidance groups that we respond to. Most of them are really very much towards the exploration end of things because they are very broad questions. The astrobiology strategy that was published in 2015, which we're now responding to, begins to narrow that down a bit. I also think there's a big difference between space exploration and space missions and how you frame what you actually do in the laboratory or in the field on Earth. And this is something that I think is sometimes lost in discussions. I just want to end on a couple of additional notes, and that is several people have mentioned today already the idea that you need multiple lines of evidence. And this is something that I made a plea for in this paper that is cited here back in 2001. And I've grown ever more devoted to that idea because life is essentially a very four-dimensional phenomenon. It is not just a bag of suspiciously complicated chemicals. It's that bag of chemicals doing things over time. And if you don't have the lines of evidence to capture each one of those, including structure, which is often put on the back burner in favor of chemistry because structure is hard to see, then I think we're missing a lot of the major evidence that we can count on. So I'm arguing that hypothesis specificity has to be really matched to the real level of understanding that you have of a particular body, a particular question, a particular set of chemistry, whatever it is. We can't simply have a blanket approach to everything. And this makes our ability to do science much greater if we actually respond to this, but it is very challenging. And with that, I'll end. Thank you. Okay. Yeah, David. I don't know where yeah. People are running from all over with microphones.
Really nice, thought-provoking talk, Penny. Your point about hypothesis-driven versus exploration-driven is something that I think about a lot, and it makes me think of this exercise that for all of us who've been involved in NASA proposals, it's sort of the bane of our existence, the traceability matrix.
Yes.
Which, in a way, is sort of a fraudulent exercise that we all do, although maybe it's a useful fraud. Mm-hmm. And I say that because the traceability matrix is where we pretend we're starting with the questions we want to answer, and then we say, given these questions, and here are the instruments we would most like to send. And we sort of present the logic that way. But of course, when you start one of these mission proposals, you do exactly the opposite. You say, okay, here's the budget that we have, and here's the mass, and here are the instruments that we could possibly send that we would like to include. Right. So let's start there, and now let's put together a story and sort of make the matrix backwards, and then we pretend we started with the questions. But it's a useful fraud because it does force us to think about, okay, what, given this complement of things that realistically we can send, what would these questions be? So I guess what I want to know is, do you think we're doing it wrong? I mean, is there a different way that we should be thinking about it?
I think that there's a big difference between the logical construct that you make of science and the temporal way that science plays out. And I think that this is one of the problems, even when you're teaching students. If you present them the scientific method, it's this logical construct. And as a student, people often naively assume that you start at the hypothesis and you march on through. Okay, no, because the universe is telling us things and inspiring us and making us ask. And so you start somewhere in this gamish of stuff. So I think that the imposition of that logical structure is not a bad thing, even if you start in a very different way. Do I think that hypothesis-driven science is flogged? Yes. And I say that as somebody who, has done a lot of exploration, that there really is a lot to be found in that synergy. Doesn't mean you just go hair off and climb mountains for the heck of it. or go into a cave or whatever one's field is, or dive into a deep lake. You have to have that knowledge of the questions in the back of your mind. But I think when you oversell the constraints, it is a work of fiction. On the other hand, fiction can always be revised. So if you actually do get awarded the funds, then you can actually go in your project where the science takes you. And I think that's one of the latitudes that we have in astrobiology to do that. And certainly the astrobiology program and our institute cut people a lot of slack. I mean, we don't make them slavishly focus on the thing they said they were going to do if something fabulous starts coming off it. So I think, good sense and moderation. And I'm just glad I don't have to write proposals anymore. Yeah, okay, Ramunis, yeah.
I don't know if that's going to be quick. I'll try. So thanks, Penny. Great talk. And I really like this exploration, putting it on the spot. And one great example is the blossoming of microbial diversity exploration, which didn't start from a major hypothesis. It was just technological advance that allowed us to now explore all the uncultured groups of microbes, bacteria, archaea, and eukaryotes. And suddenly we just, at a point where publishing another 10 or 100 new file of bacteria and archaea is annual routine, and who knows how much we still don't know. I want to now thinking about life as we don't know. We are talking about other planets. How about our planet? Can we exclude, giving all technology limitations we have right now, can we exclude that there hasn't been another Genesis and that they still proliferate in some subsurface environments?
Right. I don't know. We have always had our eyes open for this idea of a hidden biosphere of some sort. I don't know what I really think about it, but I think that keeping your eyes open for that is useful. One of the problems with it is, with our genomic techniques, the better we get at them, the worse we're going to be at detecting other types of life. And the only way to overcome that, in my view, is to go back to my earlier point, which is you need multiple lines of evidence of all different sorts. If I saw something that was very extraordinary physically under an electron microscope, I would want to pursue it. And you know that we are and have been pursuing these weird, sort of look like mesh stockings for almost 20 years now, and cannot match them up with DNA, cannot match them up with anything we understand. For all I know, they're part of the, the shadow biosphere, or maybe there's something else entirely. But they're clearly of some kind of organic origin. So, you have to take what nature shows you, and this is why I think that constantly rubbing brain cells in our community about these finds is the only way to counterbalance the fact that we have very specific, highly technically restrained, but very powerful tools to interrogate nature. We need all of those so that we can see where those are not matching up. I hope that answers. Okay, great, thanks.
Great.